Try to remember that incantation. Yeah, I can learn something too. Okay, so this is the Intro to RNA Sequencing lecture. Please do ask questions along the way. This is kind of a chance, so the goal of this lecture is to, before we really jump into command line, bioinformatics tools, R programming, Unix programming, to think a little bit about how the data gets generated and to think about how we need to keep those details in mind when we're interpreting the data as we're working through the bioinformatics analysis and workflow. So if you guys have questions or comments about sort of where the data comes from and on the sort of more of the genomic side of things, this would be a good time for us to discuss those things. Okay, so here's an overview of the course. So we're at a high level, we're going to sort of work our way through RNA sequencing. Where does the data come from? Then we're going to work through alignment and visualization techniques for that data. Then we're going to calculate expression of genes and do differential expression analysis and pathway analysis and comparisons between different methods of differential expression analysis, batch correction, those kind of things. And then hopefully we'll have time to do this alignment-free expression estimation, which is going to kind of repeat the whole workflow, but from, as I said, a different starting point where you're assuming you don't have a reference genome. And so it uses different tools, but it's like very conceptually the same idea of what you've worked through. So it sort of, again, helps you, I think, reinforce that once you kind of learn how to run some bioinformatics tools, it just gets easier to like mix and match them and pick up new ones. So the goal of the tutorial, which is all available online and which will remain available to you after this course, is to provide a working example of an end-to-end RNA-seq analysis pipeline from raw data to differentially expressed genes and significant pathways and so on. It needs to run in a modest amount of time with modest compute resources so that we're not sitting at like waiting for something to finish. So there's been sort of a few tweaks to the input data that we made to make that work quickly. And as much as possible, it's supposed to be self-contained, self-explanatory, and portable, which means we try to like provide methods for you to help you rerun this analysis when you get back to your own institutions. And we try to kind of annotate all the commands with explanation of sort of what's going on, what does this parameter do, what inputs does it take, what does it produce as output, and so on. Okay, so we'll go through like a really basic introduction to the theory and practice of RNA-seq analysis, sort of starting with just the background molecular biology. Let's see if I can get a pointer going here without crashing everything. We'll talk a little bit about challenges that are particular to RNA-seq analysis, general goals and themes of RNA-seq analysis workflows, some common technical questions that come up, which you're free to like, again, ask as we go along. And then we'll do an intro to the hands-on tutorial, and we'll be working through that hands-on material for the rest of the day. Okay, so I like to start with this, like super basic. I'm sure you guys all know this. This is the central dogma, so this applies to, this depiction applies to eukaryotes primarily. But just so we're all on the same page here, when we're talking about RNA-seq data, it's in the context of transcription. So we're starting with a double-stranded genomic template, which has many sort of regulatory features. And this sort of cartoon example of a gene, we have 3 exons and 2 introns. The introns are quite small. Many eukaryotes will have much larger introns than what is depicted here. Transcription of this thing is regulated by things like promoter regions, and polyadenylation sites. The transcriptional machinery from this template creates a single-stranded pre-mRNA molecule where the introns are still intact, and then a separate set of regulatory elements, splice donor and acceptor sites, as well as exonic splicing enhancers and silencers and intronic splicing enhancers and silencers, all work together to help the splicing machinery recognize the structure of this thing, identify introns as separate from exons, splice the introns out, and assemble a mature mRNA molecule, which is capped and polyadenylated and exported to the cytoplasm, where it will be translated into protein, and then that protein is folded and subjected to post-translational modifications and subcellular localization and other things that allow it to have its function. Probably not all of us. I think we heard during the intros that some of us are actually studying non-coding RNAs, but I'm guessing quite a few of you are interested, at least in part, in protein coding genes. So for genes that actually make a protein that functions as a protein in the cell. If we could just sequence these things, these proteins, the full-length proteins, directly and get an accurate abundance estimate of them and identify one protein from the other 20,000 proteins that might be in our cell. And we could do that cheaply and quickly and get this like really high resolution identity and quantification of every protein in the proteome for like a small number of cells that are sort of compatible with like wet lab experiments. A lot of us would probably do that, right? If we could just sort of skip right to protein abundance estimation. There is a whole field of mass spectrometry, and it kind of does do that, but it's very technically challenging and has some limitations that are sort of gradually being improved upon. But generally speaking, it's hard to get a readout of the abundance of every protein sort of from end to end out of a mass spec experiment. That remains very, very difficult and expensive and often has high sample input requirements. So many of us are using transcriptomics, things like bulk RNA-seq and single RNA-seq as a sort of proxy for protein. Looking at this depiction, does anyone want to say which of these four panels is the closest to the thing that we're actually sequencing in an RNA-seq experiment? Which one? Is it the first one, the second one, the third one, or the fourth one? I guess I've already told you it's not the fourth one because we're not sequencing protein, but which of the other molecules are we actually sequencing? Three, I see a three. Does everyone agree with that? Yeah, that's right. So we're kind of going after the mature mRNA molecules, but we're not actually literally sequencing those things exactly as depicted here. What about it is different in a bulk RNA-seq experiment? How are we not sequencing a mature mRNA? Yes.
There's a reverse transcription step and cDNA production as a result.
That's right. Yeah. So generally speaking, for the sequencing platforms that probably most of us are using, they're not actually capable of sequencing RNA directly. So we have to convert it to cDNA so that we can do DNA sequencing on the DNA version of the RNA. Anything else about this depiction that's a little bit sort of simplified relative to what bulk RNA-seq is actually doing? Yes.
Probably no fragmentation of the RNA transcripts.
Right. Yes, exactly. So we're not sequencing the whole mature mRNA from end to end, and we're not actually getting like additional like modifications like things that are methylated, or we're basically converting it to cDNA, breaking that cDNA, full-length cDNA into pieces, and then we're sequencing little chunks. And then we're hoping in the analysis that we can kind of like reassemble things or infer things about the whole transcript based on those pieces. Okay, and so this is like at a really like high level what an RNA-seq experiment looks like in terms of data generation. So we start with some samples of interest, could be 2, could be 10, could be hundreds. We generally have some conditions of interest. So we have 2 groups or more groups that we're hoping to compare to each other, or we have a time course or other variable that we're interested in. We take those samples, essentially piles of cells, isolate RNA from them. Generally like a total RNA isolation is done. We then generate cDNA from that RNA, fragment it, size select it, and add linkers that allow us to do sequencing. And then the sequencing is done in some kind of massively parallel platform. The most dominant platform right now is the Illumina next generation sequencing approach, which produces short reads. The read lengths can vary, but there's something like usually many tens of bases, like the shortest you generally see is like 50 to 75 base pair reads these days, but you might see some shorter still. And they're often sequenced in a paired end fashion where each of these fragments is sequenced from the 2 ends of the fragment towards the middle, giving us a total read length of something like 150 to 250 bases of cDNA sequenced. And if the fragment is small enough and the read lengths are long enough, the 2 reads will meet in the middle, essentially resequencing that whole fragment. If the fragment is really short, the 2 reads might actually just be identical, but reverse complements of each other. And if the fragment is a bit bigger, there might be a little space in between the 2 reads where we have a chunk of cDNA that wasn't sequenced at all that remains unknown. But we're hoping by sort of analyzing that fragment, we can kind of have a pretty darn good guess what those missing bases are and sort of know what the fragment was that we sequenced. We produce millions and millions and millions of these, often tens or hundreds of millions of these sequenced fragments for each sample that we're interested in. And then those sequences get fed into a very wide variety of downstream analyses, some of which we're going to demonstrate in this course. So I mentioned that the Illumina platform is currently dominant in terms of sort of production scale, data production. Probably many of you have access at your institute to an Illumina sequencing service or core service or use a commercial one that uses most likely one of these instruments or maybe one that's slightly older than this that's capable of producing a huge output per run. So in a matter of, 12 to 48 hours, you can produce a vast amount of data on a single or dual flow cell. So the current sort of top of the line instrument that Illumina brags about on their website, which I just checked this last night, is the NovaSeq X series, which they claim will produce 26 billion with a B reads from a single flow cell. And it has a dual flow cell mode that will give you 52 billion of those reads. Most typically in this 2 by 150 approach. And that is what our sequencing facility does. So that's become kind of the currency of all sequence data production at our center is 2 by 150 base pair reads. And because the cDNA is being sequenced as cDNA, a single run can mix an incredible diversity of different experiments. So the single flow cell could have data generation happening from 12 different labs, some of whom are doing whole genome sequencing, some are doing bulk RNA-seq, some are doing single cell RNA-seq. They could be different species. Some of them are epigenetics assays, just all piled together using the same data generation approach with barcodes that allow you to separate them out into piles and give each lab back their sequence data for their type of library creation for their samples. It's kind of an amazing approach. And this flexibility and level of parallelization has really driven the cost of this data generation down to the point that it's quite feasible to do experiments for a few $100s per sample with all of these different kinds of sequencing assays. Okay, so we're going to pretty much entirely focus on this kind of data that was generated on an Illumina platform. But I did want to just at least briefly mention that there are competing approaches that are sometimes referred to as next or third generation sequencing platforms, where the second generation is the Illumina and first generation was kind of old school Sanger sequencing. The two notable platforms in this space are the Pac Biosciences platform and the Oxford Nanopore platform. They're both amazing technology. The PacBio uses these amazingly engineered enzymes that allow you to essentially watch what is being synthesized by this enzyme and read it out as a movie. So they essentially take like a video recording of these molecules, sort of like assembling sequence, and they're able to like see each base being added, which they've achieved by like various modifications that kind of slow the enzyme down and have all kinds of really cool biochemistry that allows this to work. And it sort of happens in this little microwell where the enzyme is sort of like fixed to a surface and the molecule flows past it, and you sort of capture this information as that's happening. And then the other is the Oxford Nanopore platform, which is sort of similar in some ways, but in this case, you have an array with a bunch of pores, tiny holes, which are coated in sort of special like machinery that allows single molecules of RNA or DNA to be fed through. And then there's a detector attached to each of these tiny holes that allows you to read off the sequence as the molecule is being fed through. This is a very old idea, actually, that has taken decades to perfect or to work towards perfecting. It is still not far from perfect, but it has really started to become an interesting way to generate transcriptome data. Unlike the Pacific Bio and Illumina approaches, this one is actually capable of directly sequencing RNA, and it's also capable of detecting modifications to nucleotides. So it has the potential to give you more rich information and to allow direct RNA sequencing. It has some challenges, though. But really the main reason that people are interested in these technologies in the transcriptome context is that they allow much longer sequences. So on the previous slide, I showed the Illumina platform, just the way it works with its synthesis, sequence by synthesis approach. It's very difficult to get read lengths longer than like 250 or 300. At some point, the quality of the sequence information just really drops off. And it's not for lack of trying. They've invested huge resources in getting the read lengths to what they are. But these technologies can give you read lengths that are 10,000 bases, even up to 100,000 bases. So the appeal there is what we talked about, that maybe we don't have to fragment the RNA. Maybe we can just sequence full-length RNA molecules and just read them off and know exactly what the splicing pattern was from end to end. So there's a lot of interest in applying these technologies in that way now. It still remains a little bit challenging from a throughput perspective. So you generally get less data, even though they're longer, and it costs more. And there are some like per base quality issues. But they've really come a long way and they're starting to get, quite exciting for transcriptome analysis. Some challenges related to RNA sequencing. So this is just sort of like really fundamental things that you need to worry about or think about when you're designing and executing an RNA-seq experiment. Of course, your sample needs to be a good sample. It needs to be pure. You need enough quantity. Actually, for bulk RNA-seq, the quantity is really not that high of a demand. You can sequence a relatively small amount of RNA and get pretty good data. Quality is an issue because RNA degrades easily, and if it gets too degraded, that can cause a problem. But when we're sequencing short fragments, it's actually pretty tolerant to that. You can sequence pretty degraded RNA and get a pretty decent RNA-seq data set out of it. I would say like a bigger fundamental challenge to transcriptomics is this problem that the things that we care about, different RNA species, vary wildly in their relative abundance. Estimates are that, from the most lowly expressed but functional RNA to the most highly expressed functional RNA, we have something like 10 to the 5 to 10 to the 7 orders of magnitude difference. So you have some genes that just really don't need that many copies of the transcript to make enough copies of the protein, or the protein is really stable and you just make a little bit of it and it lasts a long time, or you just need it now and then, and so there's a little pulse of it and then it's not around for a while. All of these things kind of complicate the interpretation of the data and make generating the data for a comprehensive view of the transcriptome much more difficult, because we don't get to choose really what transcripts we sequence. It's a random sampling process. So when you're randomly sampling molecules from your pool, the most abundant things are easy to sequence and the less abundant things are hard to sequence. There are some strategies to kind of try to even things out a bit, to sort of normalize across this dynamic range so that more abundant things are kind of pushed down in terms of their readout a little bit, and rarer things are kind of pulled up. And I'll talk a little bit about that. Yes.
How do you translate something into protein?
So the question is, how readily or how do you translate transcript abundance into protein abundance? That is a good question. I don't, and an old one. It goes all the way back to the microarray gene expression days. Numerous studies have been published to kind of show for the people that are sort of advocates of this technology that, broadly speaking, transcriptome readouts do like generally correlate with protein. And then, of course, a lot of cautionary tales about where they didn't. I don't think there is really a good way other than to just kind of be cautious and to not assume that that your transcriptome readout is like necessarily like very highly correlated with, that it's only kind of loosely correlated with the protein readout. And a lot of transcriptome experiments are hypothesis generating exercises where you at some point often are kind of like going on to the protein stage and then switching to other kinds of assays that are lower throughput, but actually do give you a readout at the protein level. Yeah, good question. The other issue that is a challenge for RNAs that isn't a challenge for other forms of high throughput sequence assays, like a lot of the epigenetics assays and DNA sequence assays like whole genome sequencing and exome sequencing, is that RNAs come in a wide range of sizes. And there are small RNAs that seem to be very important functionally that get really small. So they can be a functional RNA, like a microRNA, can be as small as a few tens of of nucleotides long, whereas other RNAs can be 100,000 nucleotides long or more. And sort of like dealing with them all in one consistent assay that sort of captures everything is challenging from a molecular biology perspective. It works pretty well to get everything that's, say, above 200 bases or larger. But generally, when you're interested in those smaller RNAs, there's sort of a branching in the molecular biology prep where you create separate libraries for really small RNAs that don't follow the usual bulk RNA-seq data generation protocols. And then I already mentioned that RNA is fragile compared to DNA. And this is sort of like a follow-up to that. Many of you are probably familiar with the Agilent or one of its competitors, called the Bioanalyzer, or there are other trademark names. Essentially, it's a gel electrophoresis instrument that runs samples through a gel in a capillary so that small nucleotides come through more quickly and larger nucleotide molecules come through more slowly. There's a detector that sort of reads off these molecules as they're coming through, and it gives you a kind of readout over time. So it runs over a period of 5 or 10 minutes. And then gives you this readout that tells you sort of like what size are most of my molecules. Are they mostly of one size, or is there like a lot of different sizes, and what's the range of that size? And these two examples shown here are showing on the right a sort of perfectly intact total RNA isolation that I did from some cell lines that were just happily growing along in an incubator 1 minute, and the next minute they were being broken apart by trizol and fed into an RNA isolation. So there was no time for degradation to happen. And what you see in human, you get a pattern like this, where essentially you see two large peaks, which would be two bands on a gel that correspond to the ribosomal RNA species. And then as the RNA gets degraded, as it is in this sample on the left, which was a piece of human colon tumor that was in a freezer for several years and then thawed by me and scraped into little bits and subjected to RNA isolation, the RNA just gets broken into pieces. And so now you get what would look like a smear on a gel. It's fragmented into little pieces. And you can use the ratio of the area under the peaks of these sort of all of these little peaks relative to what it would look like if we had sort of perfectly intact RNA to come up with a score, often called an RNA integrity score, excuse me, that gives you sort of a numerical estimate of how intact your RNA is. There are some sequencing facilities that will ask you to run such an assay, or the first thing they do when you send them their sample is they will run such an assay. And if the degradation appears like quite bad, they might like hit you back with an email saying like, hey, it seems like your RNA is kind of like degraded. We generally don't like to make a library if the RNA integrity value is below 5 or something like that. And they'll often say like, do you still want to go ahead? And then you'll say yes, because you did all this work to like isolate that sample. And then you hope that your data is still good quality, but you remember in the back of your mind that there was this issue. Hopefully you remember that. And then you think about that in terms of batch effects. If there's a lot of variability in how your samples look across your cohort that you intend to do a combined analysis on for this kind of readout, that could be a problem. So this kind of like variable degree of degradation can really introduce batch effects. If it's consistent and they're all kind of consistently like this one on the left, it might be okay because you do still have fragments that are big enough to do RNA sequencing with. Okay, so unlike some of the DNA sequencing approaches, RNA-seq remains a little bit more of a Wild West in terms of the way the data gets generated in the lab. And I think this has become a little bit obscured because a lot of people now don't make the library themselves. They just send their RNA sample to a core and they do what they do. But often a lot of these core facilities will have multiple RNA data generation sort of approaches or kits that they use, and they'll have like a menu of choices you can pick from. At our center, there's like 4 or 5 choices usually, and it changes over time like quite a bit actually as kits get discontinued or people are still innovating in this space trying to make them more robust. And so then if you're comparing data from 2 different institutes, there could be actually quite a few potentially different data generation approaches in play. So it's important to keep in mind whether these kinds of variables are in your experiment and how that might influence the analysis. So this, for example, some people purify polyadenylated RNAs before making a library. Others start with total RNA and do a riboreduction step to remove the highly abundant ribosomal species. I'll talk a bit more about that on the next few slides. There are differences in the way size selection may be done with fragmentation or enzymatic before and or after cDNA synthesis. If you're working with small RNAs, there's definitely going to be some different approaches there. For samples where you have a really small amount of RNA, so you'll see this sometimes in some mouse systems where you just aren't able to get that many of the cells that you need. There are kits that will help you kind of deal with the low amount of input by doing a linear amplification. There are kits where the reads come out at the end and you know what strand was being transcribed, and there are kits that come out the end and you don't know what strand was being transcribed and you infer that information from the analysis. And there continues to be like a real mix of those two types of library data generation going on. There are some library normalization approaches, including doing a sort of exome or cDNA capture, where you kind of bias your data towards transcripts whose sequence you know by hybridizing them to this huge set of probes that correspond to, say, all of the exons of your species. And this has the effect of kind of like evening out the expression of really highly abundant things and pulling up the expression of really lowly abundant things, and kind of focusing all of your data on the exons that maybe you care about sequencing. So this is popular in a few different contexts, one of which is for heavily degraded RNA. Can really kind of rescue those heavily degraded samples, or in applications where you just really only care about the protein coding basis of your genome. Yes.
With changing the proportion then of like lowly differentially expressed genes that like falsely elevate their gene expression as well, how can that relate to the actual constant?
Yeah, so it does. And there's been a few papers sort of published about this approach and sort of asking that question, like how much does it impact the ultimate readout? And it's surprisingly not that bad, not as bad as you might think. So it does compress the dynamic range. So the highest things become a little less high and the lower things go up, but there's still like a wide range. And that range is still highly, highly correlated to like their expression levels that you would have seen if you hadn't done that enrichment step. So that is often like a justification that people will use to say, like, I'm going to use this approach. If you're comparing data where there was the cDNA capture to ones where it wasn't, then that's where you would have definitely worry about that problem, that you've kind of like, they're not on an even playing field. Yeah.
Thank you.
Okay, so this is like a little bit of just a quick walkthrough of what the molecular biology looks like when these data are generated. Always good to sort of keep these kinds of details in mind. So we start with some tissue sample or vial of cells or cell suspension, isolate total RNA from them. Generally, there's some kind of assessment of RNA quality. Back in the day, we would run RNA on a gel. As I said, now it's very popular to use this gel electrophoresis approach, and often the core will do that for you. Here's another set of examples of RNA from very intact to somewhat degraded to heavily degraded to very, very, very heavily degraded to the point where there's basically no fragments larger than a few hundred bases. But assuming you go through that and you decide your RNA is of sufficient quality to proceed, there will almost always be a DNA treatment step where you try to remove any genomic DNA that's still in your sample. And then an RNA fragmentation step and the cDNA synthesis step that we've talked about. And then if fragmentation wasn't already done, you might fragment the cDNA. If your RNA was already degraded, you might decide to just skip the fragmentation step. There may be a size selection or exclusion step, and there's various approaches that are used to effectively sort of get rid of the really small things or the really large things to kind of get all the fragments that are in a certain window, which is generally desirable for the short read sequencing platforms, to not have like a really wide range of fragments being sequenced. And then you add sequencing adapters and sequence from the ends of each of these fragments, as I showed on that earlier cartoon. Okay, and then there's often, as I said, there's this size selection step. Let me see if I can just move this, where we do RNA sequence enrichment, either through selection or depletion. And there's sort of three major categories. So you could not do the size selection, and maybe for some species that would be okay, but generally speaking, there's too much ribosomal RNA to just sequence the like convert the RNA to cDNA and sequence that directly. You just wind up like sequencing the ribosomal genes like to death and get like basically nothing else. You'd have to spend like a huge amount to kind of just keep sequencing more and more to find all the other genes. So there's almost always some kind of enrichment, and the three major approaches, which can be combined with each other, are depicted here, but probably the most popular is ribosomal RNA reduction. So you design or you order a set of probes that have been designed to match the ribosomal RNA species of your organism. You run your RNA sample on a column. The column grabs all of those things by hybridization, and you wash through everything else, and all of the stuff that comes through is sort of the transcriptome, but now you've subtracted out these ribosomal species. The sort of inverse of that is to positively select for the things that are polyadenylated. So now you're holding on to the things that you care about on the column, and you're washing away all of the so-called junk. And then once you're done that, you elute off your polyadenylated RNAs into buffer and then make your library from that. And then there's this cDNA capture approach where you're, instead of hybridizing to an oligo DT or to the ribosomal species, you're actually hybridizing directly to all of your exon sequences or other things that you've decided you care about. And you can combine the cDNA capture with the poly-A approach or the ribosomal RNA reduction. But the goal of all of these things is to sort of like help you focus your molecules to the ones that you're interested in sequencing so that you don't have to spend as much sequence data getting abundance estimates for all of the many genes that you care about. Any questions on that? Okay. Okay. And then we'll talk a little bit about stranded versus unstranded data. The example data, most of the example data for the exercises and tutorials is stranded, but there continues to be this mix where you do or don't know the strand information. We'll introduce you to a tool that will let you figure out whether and how your data is stranded. And this is a kind of like complexity that's sort of a reality of the bioinformatics workflows, that many of the tools that we're going to run, if your data does have the strand information in it, it needs to be sort of explained to the tool, how is that strand information encoded into your data? How do I tell, like, how do I translate information from the sequence data to the sort of interpretation that your read came from the positive strand or the negative strand? So you do often need to figure this out, and there's a handy tool that will kind of just, you feed some of your data, raw data in, and it'll like give you a prediction, like I think your data is stranded and the stranded information is on the positive strand, or I think your data is stranded and it's on the negative strand, or I think your data is unstranded. You can also just do your alignments or align a little bit of your data and then load your data up in IGV, and we'll demonstrate this as well. And then there's like a visualization mode in IGV that you can turn on, and then you can just go and look at some example genes, and you'll see right away, like when I have two genes in a kind of head to tail fashion or head to head fashion, I can clearly see like, oh, all the reads are being annotated as coming from this strand for this gene that's going in one direction, and they're all being annotated as coming from the other strand for a known gene that's annotated as being transcribed in the other direction. It really looks like my data has this strand information in it, because that would just never happen by chance. Or you'll see something that looks like the top left here, where the strand information is just kind of random, like there's just like a real blend of the two strands. And so that tells you that your data is unstranded. And once you know that information, you can sort of choose the right option for all of the tools for alignment and counting gene counts and other things that sort of expect to be informed about this information. And we provide a table with sort of the correct parameter setting for a bunch of common bioinformatics tools based on what your strand information is. It's surprising how often this detail gets forgotten. Like, you can ask your core what the strand information is, and they'll often kind of give you like a blank stare, like it's kind of determined by the kit, and they don't do the analysis, they just generate the data, and so they like actually don't know the answer, or they have to ask around to get the answer. Or you got data that's from a bunch of different places, and it's hard to track down who generated the data. And that's why these tools exist, to kind of like help you reverse engineer what happened after the fact. And that's why we make a point of demonstrating those. This is just like an example for cost, just using sort of our current core facility as an example. So I apologize that these numbers are in US dollars, but they give you a general idea. So this gives you a sort of an idea of the complexity. So right now, if we go to our core and say, I have 5 RNA-seq or 10 RNA-seq samples and I want to have them sequenced, this is what they'll ask us. They'll say, well, how? How do you want your RNA-seq data to be generated? Do you want to do a poly-A selection? Do you want to do one of these ribosomal depletion things? And then we have all these different options that include sort of like depletion of different molecules. If your RNA is coming from blood, you may want to include this extra globin degradation thing or reduction thing. And then we have a couple low input options in case you're really just not able to get much RNA. And then this is what we're going to charge you for each sample. And if you do a huge experiment, we'll give you a little discount. If you're doing a small experiment, we'll charge you a little bit more because there's sort of more labor per sample. And then all of this assumes that you want 30 million reads. Are you okay with 30 million reads, or do you need more? And if you need more, we're gonna charge you a little bit more. Yeah.
Sorry, just going back to the previous slide, how could you retain stranded information when in reality, like you don't, like orientation at 3 dimensions, it could be flipped or inverse. How do you like specifically know which comes from a forward and reverse one?
Can you repeat the question, please?
The question is, how do you really know what strand was being transcribed? I think the answer is that there really is a strand that's being transcribed. So you think of a stretch of the genome. It could be transcribed in either direction, but there's a gene there, like the one I showed with 3 exons, and there's a promoter at one side and not at the other side. And that promoter gets recognized by the transcriptional machinery, and it makes an RNA from one side of the 2 possible templates going in one 5' to 3' direction, a particular one. So at that moment, if you could just sequence those RNA nucleotides directly, you would know the strand, right? Because like it is one thing, and if you could read them off, you would know what the one thing was. The basic idea of all of these library kits that maintain the strand information is that essentially they just keep track of that. You make cDNA in a way that when the first cDNA synthesis happens, you kind of like mark it, or you know by the directionality which cDNA, first strand cDNA synthesis you were actually doing, and you carry that information forward through the steps. And there are a couple different ways that it's done, and the molecular biology is kind of like complicated, and the kits don't actually like explain exactly how they're doing it because it's like kind of proprietary kit stuff. But that's the basic idea. Yeah. Any questions on the sort of like different ways to generate the data, or so people often ask like, how much data do I need? That's sort of like one question that you have to decide. You can just go with what your core recommends. They will generally recommend a pretty small, what I consider a pretty small amount, like something like this, 25 million to 30 million. A lot of these, there was a very well-known paper that did an analysis, statistical analysis essentially, that showed pretty convincingly that if you have a bunch of samples to sequence and have a finite budget, does anyone here have an infinite budget for research?
Sorry.
No, I don't see any hands going up. Okay, so everyone's got like a finite budget and often, access to quite a few samples. So you have to like design your experiment and try to maximize what you're going to get out of it. And there was this really great paper that came out and said, like, you know what? You could sequence each sample deeper, but you'll generally speaking get a lot more value statistically out of doing more biological samples. Because biological noise is a real thing, and we're always kind of skimping on biological variables because of cost. And then we get results and there's like false positives, and we try to validate something and it doesn't validate. And it turned out it was just kind of like a statistical like noise or artifact of our biological system because, it was sunny one day and rainy the other day, or a different tech prepared the sample or whatever. So they made this argument that's like, if you're doing differential gene expression analysis for that analysis specifically, which is a very common one, what many of us are doing, and that's why we use it as the sort of bioinformatics demo here, you'll get more bang for your buck sequencing more samples than you will doing a fewer number of samples, but sequencing them more deeply. And that's all true. And that's where this like kind of 25, 30 million amount came from. They kind of did an analysis of different amounts of data, and they determined like for most like RNA sequence experiments, by the time you've sequenced 25 million reads, you can get a pretty good readout of gene abundance. Yes. How many replicas? That's a much harder question. Yeah, so this sort of like figuring out like how many reads will it take to kind of get like a decent readout of like most of the genes. So I've kind of detected every gene and able to tell like this gene is really high and that gene is really low, and I can detect a twofold change between my, treated condition and my untreated condition. We can kind of like model those things, and they're fairly straightforward. How many biological replicates you need is like a much more open-ended question, because it really depends on like the nature of your biological variability. And yeah, it's just, there's not a good answer. I mean, a lot of people, you do 3 to 5 and hope that that's sufficient. You'll see people that like do 3 to 5, but then within each of those they're pooling like several biological samples, trying to kind of even out some like noisiness. So it really just depends like how reproducible your experiment is. And so every like transcriptome analysis should start with some kind of analysis, like a principal components analysis kind of view of your data, where you like sort of look at the broad strokes pattern of transcriptional expression of all of your genes in each sample, and then you label each sample with different things, like your condition A that you're hoping to get compared to condition B. Do they seem to separate? Is A really distinct from B, and my replicates of A are kind of all like similar to each other, and my replicates of B are all kind of similar to each other? Or are they all kind of blending together? Are there outliers? Does it look like I've like swapped some of my samples, that kind of thing? So the question is, do you need more replicates for RNA-seq versus DNA-seq? That is a really interesting question as well. I think the answer is yes, although it probably depends exactly what you're doing. I mean, for a lot of samples, the genome in the sample is more of a kind of fixed quantity, and the transcriptome is more dynamic. I mean, the reason a lot of us are doing transcriptomics is we're interested in function, right? Function depends on context. It depends on stimulus. It depends on the environment. Whereas those cells have the same genome, the same chromosomes, the same DNA sequence in all of those conditions. Like that's more of a fixed, whereas the transcriptome is varying. And that's what we're hoping. We're hoping that when we treat it with the drug, the transcriptome responds, and that tells us something about pathway biology or resistance to the drug or whatever. So I think, yeah, I think the answer is there, yes, kind of. But there's probably some exception, but like generally speaking, yes. Yes, another question.
Speaking to replicates within cell line type experiments where you extract your entire sample, so you have whole plate, you extract all of the RNA. Is there a need for, at that point, submitting replicates where you're going to be submitting essentially just replicates of the library prep? Because it would be the exact same RNA extraction that's going
Right.
2 vials versus 1.
Okay, so the question is, imagine a scenario where you're growing cells in culture and you basically take all of the cells and put them into one RNA prep. And so you've sampled the whole biology of that experiment, essentially. Now you have this pile of RNA molecules and you're going to make libraries from it. Do I need to make multiple RNA libraries from that, like different aliquots from that one RNA isolation? Generally speaking, I think the answer is no, that as long as the input amount, like the number of nanograms of RNA that you sample from that whole prep, gives you a good molecular sampling of the sort of diversity of transcript molecules in there, which it generally will if you follow the recommendations of the sequence data generation place says give us 250 nanograms. That estimate assumes that there's a lot of molecules in those 250 nanograms, and it's going to capture most of what you're interested in, unless you're interested in some like really rare species. And because it's a solution, and the solution is like totally homogenized and mixed, that you get like a good unbiased sampling of all the molecules in there. And so you don't need to do replicate library generations. And the library generation event is generally considered to be quite robust at this stage. Like it's all being done with kits and it's not that complicated. So it's sort of assumed that like if you make a library with this kit on a Wednesday and then you make another one on a Friday, it's kind of like there's not that much variability coming from the making of the library event itself. And even less from the sequence data generation. Like in the very earliest days of next-gen sequencing, people would like run some sample on one like flow cell and then run some on another flow cell and worry about like that the flow cell was like inconsistent or something. And that's like just not even a concern anymore, at least for the Illumina platform. It's highly like consistent. So if you need to go and add more data later, that's another thing that comes into data generation. You can sequence like a little taste of your library and kind of like get a sense like how's it looking. And then you almost always have like library left over, and they can just put more of that on the next sequencing run or a week later, a month later. And you really don't need to worry about like, oh, it was a different sequencing run. The library, you can go back and resequence the library and it'll be very like consistent.
Like the flow cells, for example, if you have enough samples to fill 2 or 3 flow cells, should you stack or repeat one sample across all 3 as a read control?
So the question is if you have enough samples to fill 3 flow cells. So if you're working on the most recent instruments, I would say congratulations, you're doing a very big experiment because that would actually be a huge amount of data generation. But say you're doing that, or say you're using one of the older platforms that had smaller flow cells. Do you need to sort of like include a control, like include one sample on all 3 flow cells to kind of control for variability across the flow cells? People aren't really generally doing that. The flow cells are consistent enough that the sequencing core, they have their own strategy for how they kind of break things up, and they'll often kind of blend things together in case there are like sort of weird biases in the sequencing libraries coming from one experiment versus another experiment. And the flow cells actually perform best when there's like a real diversity of like molecules and their sort of ACGT content. But they'll kind of handle that, and they have controls that they include on every run to watch out for any problems, and they'll generally offer certain guarantees of data quality and consistency that you don't really need to worry about. Yeah. So coming back to this, the 30 million or 25 million reads, and how I was saying, it kicked off this whole discussion about replicates, and generally speaking, it's a good rule of thumb, do more biological replicates and choose sort of like a reasonable amount of sequencing for each of those samples. There are exceptions though. So this, remember I said that this paper, the focus of their analysis was if I want to do differential gene expression analysis. So they were asking the question, how many reads does it take to get a decent abundance estimate for most genes, like a kind of microarray kind of output? But that might not be your question. So maybe you're hoping to annotate your transcriptome of your unusual critter for the first time, and you really want to like make sure you cover all of the transcripts really like robustly from end to end. Or maybe you're interested in really like lowly abundant molecules, or you're interested in alternative splicing, or you want to measure the expression of specific nucleotide alleles. And so you don't just need like a count for each gene, which could be like, oh, there's some reads over here at the 3' end of my gene. I can estimate abundance from that. You need like coverage of the whole transcript length so that you can detect the nucleotide alleles that you're interested in. All of those kinds of things violate the assumption that they were basing this 25 to 30 million estimate on. So you need to think about those things when you're ordering your data so you can up the amount of data generation accordingly. The good news is that when you get up to 150 or 200 million reads, that's pretty good. So that will give you a very comprehensive sampling of the transcriptome, and will be good for most of these kinds of applications that I'm listing here, or all of them really. And it's not that expensive. But again, to the point of the finite budget, it really just depends on how many samples you're hoping to sequence and what the sort of the benefit, the sort of cost benefit analysis is. So one of the really cool things about transcriptomics and RNA sequencing is you generate this data once, and then you can answer all kinds of questions of it. Particularly if you've generated in a deep way, you can do gene expression and differential expression analysis like we're going to do. We actually have an alternative expression analysis section that we deliver in a sort of longer version of this course. You can do transcript discovery and annotation. You can do allele specific expression, detecting allelic bias, loss of heterozygosity. You can detect somatic mutations through mutation discovery. You can do gene fusion discovery. You can study RNA editing and so on. All of these workflows have some common themes though in terms of the bioinformatics analysis. They basically all go through these steps of starting with obtaining the raw data. You may need to convert it from one format to another, usually not. And then some kind of alignment or assembly of the reads against something known about your species, the reference genome or transcriptome, most typically. And then process those alignments with tools that have specific goals. So the alignment file becomes kind of a one-stop shop, and then you can feed it into various downstream tools that ask different questions of those alignments, that try to assemble short reads into long transcripts, to try to study alternative isoforms, that try to detect mutations, that estimate the abundance of each gene, and so on. And so there's, a whole bunch of tools from many labs that like ask interesting questions of RNA-seq data, starting with a BAM file as input. That's the alignment file. And all of those tools, that's where things get kind of wild, is the output of all of those tools is, quite, bespoke. So then usually there's some kind of downstream step that's like, okay, every one of these tools outputs the information in a sort of different way that's sort of suited to that question or interpretation of that kind of analysis. And then there's some kind of post-processing where you feed those output files into often R or Python to visualize the data, maybe format it or organize it or integrate it with other data in some way. And then generally some kind of summarization and visualization where we start to do statistics and comparisons and create figures for papers and so on. So there's a ton of RNA-seq tools out there. There's many different RNA-seq data analysis workflows for differential gene expression implemented in many different kind of analysis platforms and languages. This is a depiction of some of these tools, starting with sort of alignment at the top and going down to differentially expressed genes or transcripts at the bottom. About half of these tools are covered in this course, and the others are, conceptually very similar, have different commands, but if you can figure out how to run, say, Callisto, for example, you can figure out how to run Salmon. So we're going to go through this workflow on the left here, starting with HiSat alignments. HiSat and STAR have really become the two most popular aligners. They produce almost identical alignments. And then we're going to use HT-seq for counting our reads against known genes. And then we're going to use EDGE-R and DE-seq for differential gene expression analysis, and then we'll feed that into the pathway analysis. We're also going to do a kind of parallel analysis that uses string tie and ball gown, which, as opposed to the sort of basic count information, actually gives you sort of transcript assemblies that you could use for sort of isoform-specific expression analysis. And then the reference-free approach that I mentioned uses Callisto and Sleuth to do the transcript abundance estimation and differential expression analysis. Okay, so I wanted to end this lecture with a discussion of bulk versus single cell RNA-seq analysis. Many of you indicated that you're interested in both. A decent subset, you're actually taking the single cell course, which immediately follows this one. So I thought it's just like, as we think about the bulk RNA-seq, let's just keep thinking about how this differs from single cell RNA-seq and sort of what impact that has on the way we do the analysis and the way we think about like how we would use these for our experiments, when we would use one or the other. So does anyone want to volunteer a difference between single cell and bulk RNA sequencing, maybe starting with the most obvious difference? Could be, there's lots of different categories of differences though. Yes.
Or here, you can raise your hand and I'll feed it over for you.
Yes, please interject.
Shout it out.
Yeah. Okay, go ahead. Yeah.
First question that I was asked, like, why do I want to go into single cell instead of like the bulk RNA sequencing? And it's because I'm interested in the cell types that are in a heterogeneous population. So I'm working in a very complex organ. The inner ear has a lot going on, and we want to know what different cell types are doing. We're proposing that there are stem cells in the inner ear, so we want to know how they're behaving differently, and that's why we're choosing single cell instead of having like the bulk giving us like the overall picture.
Right, yeah, so that's sort of like the obvious sort of like rationale, and sort of the dream of single cell transcriptomics is like there's a lot of biological systems where we have cellular heterogeneity. We have a mix of different cells with potentially like quite different gene expression profiles, all in this, working closely together. You isolate a sample and there's a mix of them. And if you just take that bulk sample and do bulk RNA-seq on it, you're getting a very blended view of the transcriptome. All the signals are kind of mixed together, and you sort of get this, like, people use different analogies, but like the blender one is like you put a bunch of like different fruits into a blender and you blend them all together and you get a taste and you can kind of taste the blueberry and the pineapple or whatever. But it's hard to like isolate the blueberry and the pineapple essence of the smoothie after they've all been mixed together. And there was like a whole subfield of bioinformatics that was going on for years where they would like take bulk transcriptome output and try to deconvolute it. So there's various deconvolution tools that will basically try to say, okay, what if I make certain assumptions or guesses, and then I refine from that point and I try to like separate out the signal into different piles. So that's possible, but really hard to do well and has just like fundamental limitations. You can also try to like in the lab, you can try to separate your cells by purifying them or using fact sorting, other things. That's great. That can work really well, but it sort of creates like a bigger burden on the upstream part. And there might be cells that you just don't know how to separate well or difficult to separate well, or in the process of separating them, it's just more manipulation of your sample. So you're kind of introducing bias or maybe causing like RNA quality issues because it takes so long to do that. So the beauty of single cell is you just take all your single cells and you feed them into this box that creates all these individual sort of droplets, and you're able to then get like a transcriptome readout for each of those individual cells, and you can then kind of put them in piles and say, okay, here's my rare stem cell population, and now I'm getting like this amazing like cell by cell readout of what's going on in those cells. So that's the upside. But what are the limitations maybe?
Culture, because you have to be really specific about the type of cell that you are interested in, whereas, you know, if I take a biopsy, there's a bunch of different cell types in that biopsy that I took from somebody's muscle.
Yeah, so the comment was, does it like kind of depend on culturing? Yeah, you may have like a biopsy sample that's just really mixed, and then what do you do about that? Yeah, I think that's true. Like for some samples, it's just not feasible to like isolate them or to be able to grow them. Like you need to be working with primary tissue, and the primary tissue has important heterogeneity that you want to capture. So you're using single cell for that. And that's sort of like one of its areas of strength. Yeah.
Probably one of the issues too with single cells, the lower throughput, especially with the RNA abundance, since you're looking at it from a singular cellular level. So even genes that were moderately expressed, because you did bulk with many cells, you could still capture some differential gene expression that is somewhat changing, but especially with cells like single cells at a time, it might be harder to have that. So you might have less of a dynamic range of differential gene expression that you can look at from your results.
Yes. So the comment was that in single cell, you sort of have kind of like you have single cell level gene expression estimates, but the readout is much lower. So like genes that aren't really abundantly expressed, you're not able to detect that well. Even a moderately expressed gene, you might be able to get really good differential gene expression analysis from the bulk approach. But when you go to single cell, you just have like a few reads. And so it's hard to tell like the difference between like condition A and condition B, or sample type A and sample type B, cell type A and B. Yes, that is true. And that's probably like arguably the biggest limitation, I would say, of single cell. Like when you first start working with single cell data, you might have like an expectation for like how like detailed the transcriptional readout is, like how many reads you're getting for each of your cells. I would say like, and then you start looking at the data and your like expectations drop like a bunch. But probably for most people, they need to like drop their expectations like again another order of magnitude. Like when you start to really dig into single cell data, it's amazing how sparse it is. Like it's really, really, really sparse. It's basically all nothingness with like a few sprinkles of information. Like you have this massive matrix of cells and genes and counts for those genes in each cells, and the counts are just small. They're really small, and there's lots and lots of zeros. And you know, for some applications in RNA-seq, like if you have like some even small counts for 2,000 genes in one cell, that's still like amazing. And you can do a lot of really cool stuff with that, but it's just like really important to keep in mind that limitation, that the data is very sparse. It'll be biased towards things that are more like abundant and are therefore easier to sample at this relatively low level of sampling. And if you just look in like the QC outputs from your experiment, you can see like how many reads did I get for each of my cells and how many genes were detected. And then it's simple math. You can say, well, how many counts would that be for each gene if I just did a simple average? And the numbers are, you know, it's often quite low. And the data was already really expensive to produce, which is another difference between bulk and single cell. And unfortunately, often, even if you have a lot of money, you can sequence more, but at some point the library becomes saturated, and even at saturation for most single cell RNA-seq libraries, it's still relatively sparse. You can like produce 1 billion reads or even 2 billion reads for a single cell experiment, which will cost you quite a bit, and you still have pretty like sparse counts of most genes except for the more abundant ones. Yes.
Could you give an example maybe of where a single cell approach might be best versus where bulk might be the best versus where a combined approach would be ideal?
Yes, I think maybe that's a good way to end this section. So the question was give an example where single cell maybe would be the best, where bulk would be the best, or where you would combine them. As like a kind of like data science nerd type person, it's really hard for me to avoid saying like just do both all the time, just because, but you know, that's expensive. But they really are complementary. So they almost, I would say, like very often will like complement each other. Examples where you would want to do bulk RNA sequencing, I would say if your sample is already low heterogeneity, like don't just do single cell because it sounds cool. If you've already purified your cells in some way, or you're able to like macro dissect them in some way, they actually don't have that much heterogeneity, then you maybe aren't going to get the kind of like the biggest sort of fundamental value of single cell. And you'll be able to get a much richer picture of the transcriptome, which with much more accurate gene abundance estimates for many, many more genes by doing bulk RNA sequencing compared to single cell RNA sequencing. And then you can kind of reverse that logic and say, well, if I have a sample that's really heterogeneous and actually the cells I'm interested in, this cancer is like a perfect example of this. We've done so many transcriptome experiments over the years from bulk tumor tissue samples where we're just like, we isolated a chunk of tumor and we did our bulk RNA-seq or microanalysis, and we looked at the output and we said, that's the expression of the tumor cells. Like that is the thought in our mind. And then there's like this asterisk, like, oh, there might be some heterogeneity. Like maybe that's a worry that I'm just gonna like dismiss and then move on with making grand conclusions about my data. And now that we've done all these single cell experiments, we realize like all those papers, all of those years, like only 20% of their cells or only 30% of their cells were actually tumor cells. And they were like thinking of that data as like the signature of the tumor, when actually it was a blended signature of 20 or 30% tumor cells, and then all this other stuff, which by the way was varying from sample to sample, creating a very, very muddy picture. So the readout was like not what we really thought it was. The data is still useful, but it's like kind of really important. So if you have that situation where you already know in advance, and this is often the case, that there's a mix of cells going on, then that's when single cell data becomes really important. If you're working in a system where maybe your transcript annotations are not that great, that might be a good situation where you do like a 2-phase experiment, like you do some isolations of different cell types, you do really deep RNA sequencing on those, and you get like transcript models. So now you kind of figure out what like the important transcript structures are, what's there with this deep version. And then you go back to your heterogeneous like biological samples that are sort of like that you want to be able to just isolate and sequence directly, and you do the single cell and you kind of combine them that way. Yeah. Those are just a few examples, of course. Yes, there's a question at the back.
Yeah, sorry to kind of throw a wrench into this, but what about like single nuke RNA-seq instead of single cell? When would you preferentially use one or the other?
Yes, the question is, what about single nuclear sequencing? When would you do single nucleus sequencing instead of single RNA sequencing? I think our experience has been the most common reason that people do single nuclear is just like a technical one primarily, that if the sample like workflow is such that you just can't get fresh sample that you can feed directly into a single cell experiment. Like if you're working with model organisms or cell lines or something, maybe you go straight, it's relatively like convenient to go straight to single cell. But if you have frozen samples and you have to thaw them, many of the cells won't survive. And so a lot of like single nuclear sequencing is being done in that context where there's a sample quality issue. And then if you're using the 10x platform, there is now a FlexSeq, which is relatively recent, that involves like a fixing stage that makes the sample prep even more robust, so working from frozen or even FFPE. So I think it's mostly that kind of like data sample quality kind of concerns. But if you're particularly interested in like the combination of RNA and epigenetics, there's also the multi-ohm sequencing that does single nuclear with single cell attack-seq. That works off of the nucleus approach, so you might do it for that reason. So I think there's a couple different reasons, yeah.
Sometimes also the example, but could be in a different sense, but like you have like cells that are really tightly that are all part associated. You might still bust them up.
Yeah, so Obi just added the point that like there's also sometimes like cell-cell interaction reasons that like there are cells that are difficult to dissociate from each other without like damaging the cells anyways. Whereas the nuclei might be more robust. Like if you do the homogenization to the level of nuclei, you're kind of getting back to more like the single nucleus that was in each single cell. There's a similar argument in some cases for really fragile cells where the whole cell just won't survive. Like certain cell types are really big, or they're just like, they break apart really easily. And so they're not going to survive the single cell approach anyways. And so those cells wind up being missing from your single cell data output. And then you do single nuclear and you're like, oh, there they are. Because it's just kind of like a very different kind of workflow for the samples. So we've had, great results from both in different contexts. So yeah, it can be worth just kind of trying, do a pilot experiment where you kind of try both ways and see sort of what it looks like. Yes.
Applied this single nuclear approach to multi-nuclear cells, like single cells, and has there been successes with that, like a placental cell or osteoblasts or?
We haven't. So the question was, have you applied the single nuclear approach to cells that are multinucleated? Not sort of like on purpose, although we have done some experiments with Reed-Sternberg cells from Hodgkin's lymphoma that are multinucleated. And it's not really clear, like it's not easy to tell from the output, like are we getting double nuclei like output? I think the baseline assumption is that the nuclei will be separated maybe, but the binucleated cells will be more likely to form a duplicate, but we haven't been able to really quantify that convincingly. I think it's tricky. It's tricky to retain multinucleated versus mononucleated information but I'm sure there's someone out there who's looking at that specifically. I would be interested to see. Yeah. I think are we up on our break? It's time for break. Okay. Okay, so I think that's a good time to stop. So that was the end of the discussion, and we'll start the next section by just kind of going into the website where we're going to walk through all the hands-on material.